All right. Hello, everyone. I hope you're well. I'm Parisa Hosseinzadeh, an assistant professor at Knight Campus, University of Oregon. And I'm super excited to talk to you about developing a novel In-Silico tool for heterochiral macrocycle design. And I should also note that I had a stroke 2.5 years ago, so I speak a little slower. Projects in my lab are all really exciting protein-based projects. I design peptides all the way to try to mimic new enzymes. But today, I am going to focus on the projects that are related to peptide modeling and design. And in particular, I want to talk about developing a new in-silico tool for heterochiral macromolecule design, HRAS, binder design, and then developing a novel tool for L-only macrocycles. And if you have any questions about other areas, I can answer them at the end. So antibodies and small molecules are key therapeutics that come to my mind when we talk about disease. Small molecules like acetaminophen dimer that's shown here are great at permeability and also interact very well with deep pockets. But they're weak at targeting flat surfaces and they're really hard to change. For example, you can look at this one. If I want to change it to S, it may be not possible, or maybe I have to go from a start to find this. Antibodies, at the other hand, like this IgG2A monoclonal antibody, are very well studied. They have very good selectivity targeting molecules. But unfortunately, they're not permeable. Also, and it's hard because there are a lot of molecules that need to be targeted when it is inside the cell. Also, they need to be injected, which is hard, and some people are afraid of injection. So there must be a third option. And I would say that peptides are attractive therapeutics that are complementary to small molecules and antibodies. In contrast to antibodies, they can be cell permeable. They have the ability to be made out of non-canonical amino acids, and they're also made by solid phase synthesis, which is more robust than synthesizing it with a cell. In contrast to small molecules, they have the ability to bind to flat surfaces, and also it's really easy for them to change one small amino acid to another. So for example, if you want to change alanine to leucine, you just change, the backbone is the same. So the changing is really easy. In contrast to L and linear peptides, cyclic peptides also have some advantages. They are more rigid structures, so they can help with binding. They're more stable against proteases. And also cyclic LD peptides, the green ones that you can see here, have more diversity than just L peptides that you can see drop here. The first paper by myself and others showed that designing cyclic LD peptide with three predefined conformation is actually doable. One of the heart of this paper was that we used GenKick, which is a stochastic sampler. So it has an anchor, and then it has 3 pivots. At the first, they perturb the degrees of freedom of all these orange ones. What happens at the second step is that the green ones that are pivots, the degrees of freedom becomes calculated, and then they finally find this structure. Sometimes this calculation here results in no answer, so that's why it's called stochastic. Using the method, we actually designed multiple peptides that bind to targets with high affinity. I designed anchor extension myself that controlled some of the binding to HDAC, and then one of my postdocs actually designed peptide drug conjugates. But there are still lots of issues here. GenKick, as I told you, is a stochastic, which is good when you're trying to sample something in space without anything else from it. But it's not great when you want to design for a specific target. It's also very, very slow, very slow. Crystal structures are not always as we designed, and it's partially because of the sampling problem that we have, that we don't sample all of the conformations. And then permeability is even harder. So really, we ask this question of how can we solve these challenges? Bharadwaj et al. In 2016 actually designed cyclic peptides. The peptides have to have a disulfide bond, and also it's L only. Then I designed cyclic peptides that are heterochiral from 7 to 14 amino acid. And then Solveson tried to design formers because GenKick actually can't be worked on formers. And they showed that it can design alpha, beta, and gamma amino acids. It was also the time of molecular ML and DL. So the lab of Lin et al. Showed that we can actually transfer long molecular dynamic simulation to something they called a stream to make it much shorter. And then the lab of Bharadwaj showed that they can actually make 20-ish residues via deep learning. But the problem is that that's only L amino acids. So we knew that the middle of macrocycle L and D peptides are something that we must address. And that's when Andrew came in place and said, I'm going to try to design LD heterochiral macrocycles by deep learning for sizes 6 to 15 mers. So first, we need to have a huge group of cyclic peptides, which unfortunately we don't have through PDB or NMR. So we need to actually design these peptides, and we chose GenKick. because it's one of the few methods that can create mixed L and D amino acids, and also we already had the software, so it made sense. So we sampled peptides by using glycine as the amino acid, because glycine is agnostic to L versus D. We generated 50,000 8-mer macrocycles using glycine, and then we explored its Rosetta energy as well as QM energy. To permit efficient training, we wrote an application, extract degrees of freedom vectors, which actually converts structures to a binary database. So the features are torsion angle, dihedrals, bond angles, phi, psi, and omega, bond length, and also energies of each structure, both Rosetta energy and then QM energy. After that, with the help of Dr. Mulligan and Dr. Renfrew, we developed cyclic K, which is an encoder-decoder. In essence, the cyclic K takes all the possible phi, psi, distance, and angle, and the hedral values to generate a degrees of latent space, and then from this latent space it can then go back to the almost exact peptide using decoder. The good thing about latent space is that although it's much shorter and lower dimension, it still gives us the ability to do energy optimization when gradient descent can be done by the latent space. So we ran it on cyclic K. So epoch is the cycles that it goes through. It's like time. And then loss means that lower the loss, more accurate backbone reconstruction. An ideal cyclic K, which you can also see here, loss curve shows a smooth, steadily decreasing training loss that is shown here. And it actually is true for 6-mer, 7-mer, and 9-mer, and we're on the way to trying to do 10 to 15-mer, so stay tuned. So if you look at the backbone here, you can see that it increases linearly with the size from 2 to 3 minutes, but it's 3 minutes per 5,000 generating structure. So it's pretty good, I would say. Cyclic K scores generally better than Genkic, as you can see. And also, cyclic K have more backbone H bond. So instead of 1 to 2 with that Genkic makes, cyclic K makes in average 4, which indicates is higher stability without binder. We then wanted to know if we can do in painting. So in painting means generating the rest of a cyclic peptide structure around a fixed motif, which we call it anchor, which ensures that it preserves the portion that stay exactly where you want it. At the same time, and also I should say that any number is acceptable as long as they're in order. So we tried that, and it worked really nicely. So you can see that the anchor or the motif are here, losing less than 1 angstrom, whereas the scaffold is over there sampling a lot of things. We also tried to see if we can have two discontinuous motif that was not possible before. So imagine that R is the first one, and then through a vector of t, it generates the second residue. And we want these two residues to be always the same, whereas other residues can move. And I put 2, but it can be 1, it can be 3. This doesn't matter that much. So Andrew tried to tweak that a little bit, and he managed to get to the position that the orange residues, which are the ones that are anchored, are constant. And I should say that this is the results is much nicer now. Then I wanted to see if cyclic K actually is interface aware shape. So Lennard-Jones potential has the lowest minimum that is dependent on the environment. And lo and behold, you can see that the peptide conformation produced by cyclic K depends on the geometry of the binding interface that is conditioned on, which was really amazing. And then finally, we showed that our model is not just hallucinating shapes, it actually learns to reproduce real macrocycle geometries seen in nature, which is very amazing. You can see 3 of them here, and we have some more that is exciting to watch. When this method was developed, Andrew and an undergrad thought about, can we show the power of this method in generating inhibitors against mutants of HRAS? This was really amazing because cyclic K is the first practical AI system that can generate realistic cyclic peptide shapes and sequences. So we wanted to see if we can also make binders. So we chose HRAS. HRAS is a GTPase that's central mediator downstream of growth factor receptor signaling. Mutations in HRAS can involve in many cancers, such as non-small cell lung carcinoma, head and neck, bladder, and thyroid. Unfortunately, for many of these mutants, there's no current medication. So the question was, can we use cyclic K? And I should also mention in here that GenKick never worked for us. So we decided to run cyclic K. We have this tryptophan anchor that is bound to not the primary active site, but the secondary site that these amino acids take care of. So we use this as an anchor. And then we went through and ran a cyclic K, designed it, filtered it, and folded it. And then lo and behold, 14 HRAS designs had p-nears of 8 or above. And p-near is basically saying how likely it is for you to fold to your native structure. Then we decided to try to see how it reacts in response to molecular dynamics, showing the peptide control kinetically. So this over here is just glycine. It should shoot out. It should have not good binding. So we run it as a control. And you can see that many of them are good, but hey, some of them actually are not that good. And it's great that we run it because they're not visible in Rosetta. Then we decided to use another method of molecular dynamics in which we apply some force, and then you decide when this force is going to result in separation. So you can see that some of the peptides are actually getting separated similar to amino acids, but some of them are staying tight, which is absolutely fantastic. And I should say that the value here starts at 2 because it's between the center of mass of this peptide and then the center of mass of this peptide. So that's why. So it's really exciting. We have some peptides that we want to test, and it's really cool to see what the results are. Then, while this was going on, a postbac and graduate student said, can we repeat the same success by developing a more accurate cyclic MPNN for L-only cycles? So, why L-only? It's cheaper to order, it can be made in bacteria, and it's also more similar to proteins, so we can use all the tools that are available in proteins to improve this. Since we are running it in proteins, we are going to use alanine Ramachandran instead of glycine that has both sides. And we are going to build cyclic MPNN. The first step is, of course, generating 10,000 backbones using cyclic MPNN, and then designing them using protein MPNN, folding it using high fold, and then calculating RMSD, and if RMSD is lower than a threshold, going forward. So we have made cyclic MPNN, we had protein MPNN to compare it to, and then we also had high MPNN, which was another deep learning method that claimed to be good. So we took this as well. As you can see here, cyclic MPNN is doing as good as protein MPNN, if not a little better, and high MPNN is off, I should say. For the recovery, cyclic MPNN recovers between 40 to 60%, which is more normal. Peptide MPNN for some of them has very high recovery, and I think it's because those are written in with protein MPNN, honestly, and then high MPNN is very low. And then if we check RMSD, we can see that cyclic MPNN and protein MPNN are all both good. Cyclic MPNN is always a little better, and then this one is not great at all. And you can, I hope, see it better in 14-mer space that cyclic MPNN and protein MPNN are basically in the same region, whereas high MPNN is searching somewhere else. We try to also see how good it is in RMSD as well as PLDDT, and PLDDT is just a measure of how well the peptide is similar to the design. And you can see that in all of them, RMSD is lower and PLDDT is higher. And you can see some of the results here, and then some of the problems that protein MPNN shows. And it's true in 14-mer as well, that cyclic MPNN has a better PLDDT and lower RMSD. With that, I would like to thank you all for your attention. And I like to thank funding, thank my students, all the awards that they won, thank to my collaborators, and I'm very happy to answer any question that you have. Thank you.